Thanks, Victoria. Good morning, everyone. My name is Neil Foster. I'm here to talk about some of the work that we do in Work Package 5 for Pro Health, which Sandra introduced yesterday. Specifically, I'm going to talk about the gene work that we are doing at Nottingham University on the Pro Health project. As you all know, the production diseases that we are looking at on the Pro Health project are multifactorial. Can be many, many factors which actually lead to a production disease. We have factors which are due to the sow. We have many different management factors such as stock density, tail biting, infectious diseases occurring. We have ultimately production loss and animal welfare. The point here is, though, which Sandra actually mentioned yesterday, is that we can have an infection present. We can have some sort of pathogen present, and we quite often do, but there can be other factors such as environmental factors which then take that towards a disease and production loss. This is a slide from one of the Pro Health newsletters that was produced in November 2015, which Jarko Nurmi, which I think he's the last speaker today, will probably talk a lot more about. But what you can actually see from this slide is very interesting, because what it shows is that the cost of production disease can be as much as 30 to 40 euros per fattened pig, which really is a tremendous amount of loss. And you can see the various factors on there why. You can see the various factors on here, on there, why we may get production loss in sort of for individual cases. So parasitic infection, for instance, can lead to something like a €7 production loss per fattened pig. If we add all these factors together, then it can be as much as 30 to 40 euros. So production disease is very important and something that we need to keep sort of studying, really, because we really know very little about it. Okay, so how do we test for disease? Obviously, we have serological analysis where we will look at things like antibodies. Antibodies, though, do not necessarily tell us anything about current infectious status. So, for instance, if we have an IgG response, which is quite often looked at in the blood by an ELISA analysis, all that really tells us is that the animal at some point has more than likely seen a certain pathogen. It doesn't mean the pathogen is there at the moment. We can look at things like cytokine analysis, which a lot of people have tried to do, to try and sort of look at the cytokine analysis in terms of different diseases and magnitude of disease, whether these are subclinical or clinical. But this has been quite unsuccessful, really. So some studies have shown that certain cytokines could be important biomarkers, and then other studies have shown that in different situations with the same disease, they may not be. We can culture a microorganism, because of course we do that. We can say definitely that this animal has an infection with a certain microorganism. But that's not really as easy as it sounds. I know that the veterinary practices quite often, not just the ministry or whoever, government practices, but the actual small practices which go to various farms, will quite often culture and look for specific microorganisms that they may suspect as being common, such as E. coli, for instance, in pigs. But there's some microorganisms that we can't really find. They're very, very difficult to detect, and in some cases they're impossible to culture and grow, or else it takes a very, very long time for them to culture and grow, and we quite often need information fairly quickly. So again, microorganism culture, although ultimately it tells us that the animal is definitely infected with that microorganism, it can be quite difficult to do. Okay, so how can gene analysis improve that situation, the current situation? Probably most of you are aware of PCR, polymerase chain reaction, and PCR can be used to very rapidly detect DNA or RNA from microorganisms, and we can quantify the differences between, for fluorescent signals using quantitative PCR by measuring the differences in fluorescent intensity. So we can actually see the difference between a subclinical infection and a clinical infection. We can look at levels of different pathogen. So QPR is a very, it's very accurate, it's quite quick, and it's a very good way of assessing infectious status. PCR we can also use to very rapidly determine between different strains. So I said that, or different strains or different pathogens, but I said that some pathogens are difficult to grow, take a very long time to grow. And with PCR we can rapidly find these, detect them, and say what strain of that pathogen is present, as long as we know what the genetic signals look like, and then we can take that to use in diagnosis. Another way that we can look at this, though, is that we can, instead of looking at the pathogen itself, is the pathogen there, and what strain is there or whatever, we can also look at what gene changes occur in the host during a production disease. Now, the good thing about that is we can get a production disease, as we said, it's multifactorial. There may be very low levels of a pathogen present, and there's all sorts of other factors which are then causing the production disease. But you would expect that those other factors would be represented, would be able to be detected within the changes of the gene of the pig. And that's essentially what we are doing at Nottingham, we're doing in ProHealth. We also have these new platforms which have come on stream in the last few years, where we've got high throughput PCR, such as Biomark. And this can rapidly analyse something like 10,000 different probes very, very quickly. So we can actually analyse massive amounts of data using PCR now. Okay, just a quick sort of reminder about what PCR is about. We take an RNA primer. First off, we convert the DNA into single-stranded DNA, and then we use an RNA primer in conjunction with DNA polymerase. We then get extension of the RNA primer, a DNA synthesis, and it's this that we actually measure. And in the old days, we used to actually put this on a gel and measure it on a gel. But nowadays, we don't do that, unless we actually want to take the product off. So what we do now is then we use same sort of technique, but instead of measuring the product on a gel, we'll actually incorporate some sort of reporter dye. So here we've got this one here at the top, where we've got the use of cyber green. And if we incorporate it, we've got the RNA primer and the DNA template, and we add cyber green. Cyber green will stain double-stranded DNA. So essentially, as long as that primer has recognized something and it's now extending, the double-stranded DNA will be stained by cyber green, and the amount of cyber green that we've got, the fluorescent cyber green that we have, tells you how much product is actually there. So now we start to really quantify. On the old gel systems, it was really difficult to quantify, and in some cases really impossible. But now we start to get to a level where we can actually quantify by differences in the amount of fluorescent signal that we get. We've even moved on from that now, and so we tend to use these probes, such as in TAC polymerase and TAC probes, where we actually have a probe and a quencher system. As long as the probe and the quencher system are close together, the quencher stops the probe from working. So basically what then happens is, as we get multiple extensions, the quencher, rather the probe, drops off. And so therefore it's now moved away from the quencher, and it can then fluoresce. Now these can be very highly specific. They're very specific for the DNA sequence. They cost more money, but they are very good. And again, what we are measuring in this case is the difference in this fluorescent signal that we're getting. But this is quite specific now. And it's on this that the basis of a lot of the modern techniques that we use actually work. It's really on this principle. Okay, so work package 5 for ProHealth, which is the one that I lead. The University of Nottingham, we are using and a microarray, which I'm going to talk about in a moment, where we actually measure 44,000 genes within the pig genome. And we look at the changes in these genes with production disease that we have. At INRA Tours, they used this high throughput PCR, fluidyne system, and they actually measured 96 specific immune genes, again within production disease. At VRI, Czech Republic, which Ivan is going to talk about next, their group has been using Illumina-Seq, which is a deep sequencing technique, and they've been actually looking at changes in the biome by variable genes within the bacterial 16S RNA, and he'll tell you a lot more about that. So ProHealth essentially is looking at production disease, but it's looking at from the molecular aspect, whether it's the host or whether, in this case, the last example, whether it's actually what sort of microbiome we have in the intestines or the lungs of pigs. And there's a great deal of interest in that now. And there's a lot of information now to suggest that we can actually look at, I don't want to preempt Ivan's talk, but there's a great deal of information now to suggest that we can actually look at changes in the biome, the microbiome within an intestine, for example, and in some way we can actually relate this to disease or condition of the pig, or other animals, in fact. Okay, so the microarray. A microarray, essentially, we have a piece of kit that all of these spots are on, which is the size of a microscope slide. So on this microscope slide, we have printed 4 times 44,000 oligonucleotide probes. So you can see here that we have, this is a typical slide here. So we have, that would be the same, these are the same, but this one we could have test there, and we could have control uninfected or no problems there, and we look at all the same genes and we replicate 4 times for each one, and we can do that on for 44,000 genes on one microscope slide size piece of kit. Essentially how it works, again, is a similar sort of idea to what I talked about, how we use fluorescence to measure, to quantify disease or various aspects of the genotype. So here we've got a fluorescent probe. First off, we have the microarray printed. So these are the oligonucleotide gene probes, which are throughout the microscope slide. We then take our genomic DNA and we add a fluorescent probe to that. We then have to hybridize these, and then we read the strength of the signal, which is correlated with the amount of oligo and genomic DNA interaction. So essentially, there's a washing step in between this. So essentially, the amount of signal that we have here at the end tells us how strong that interaction was. And we can then compare, between controls or between different tests. So that's basically how the microarray works. We use this system called the TCAN hybrid station, and this system we can handle up to 4 slides or 16 arrays simultaneously, so we can go through quite a massive data set very, very quickly. Another good thing about this system is that it's very sort of hands-off, if you see what I mean, in that you don't have to really do a great deal, and the less we interact with these things and possibly contaminate them, the better. So it's a great system for cleanliness, if you want to put it that way, making the microarray much more accurate. Okay, so first thing, something that I should have actually mentioned was that what we did was, to begin our microarray, we got samples from various partners across Europe, and the first thing we had to do was to create a standard operating procedure, an SOP document, which went out to all partners which would send us samples, because we had to make sure that the samples we got back were taken from exactly the same place, whether it was the intestine or whether it was the lung. So we got an SOP together, which everybody read and sort of took care of, to ensure that the samples we get back, so if we're saying this is what lung tissue looks like, we're talking about the same area of the lung. If we say this is what intestinal tissue looks like in terms of its genomic changes, we're talking about the same area of the intestine. Very, very important thing to do. Another thing that we had to do was to get sort of sample packs together to send out to all of our partners, and the sample pack, as well as including the SOPs, also included RNA later, which is essentially a fixative for RNA and DNA, and that allowed the tissue to be immediately fixed when the animal was killed. One of the problems of a project such as this is that lots of people could say, well, we can go to the slaughterhouse and we can get as much tissue as you want in the slaughterhouse. We can have the tissue diagnosed, so we could say it's that pig had pneumonia or that pig has some sort of problem, we can get even that tissue. The problem is, by the time the animal is killed and it gets off to a point where tissues can be taken, the RNA and DNA has degraded to such an extent that we can't really get anything worthwhile from it. So we have to make sure that when we get the tissue, it's immediately put into this fixative. Once it's put in this fixative, it can be kept for quite a long time before the courier then brings it to us at Nottingham. But that step is very, very important. And to make sure that was always done, what we always did was we would test batches. So we get the samples in and we test the batch for DNA quality. And we do this in a number of different ways, but if we were unsure, so if we use a certain technique and we were unsure about it, we would actually just run this off in a DNA gel, and you can quickly see if the DNA is degraded or not because it looks all streaky and horrible rather than nice bands. So we always tested the samples as well before we went on to use them. Okay, so the first study that I'm going to talk about, these studies are all still ongoing, but with some piece of work that we've done, we've got further than us, I'm sure you can understand. So I'll talk to you about some of the studies that we've done, tell you a little bit about them. So this first study, pigs were diagnosed with pneumonia versus healthy herd controls. So you can see here then, this is the microarray, this is what the microarray looks like. So on the left we have the healthy controls, and on the right we have pneumonia. So what you can see from that is it's just a massive green spots. Now essentially what we're going to do is to measure how those green spots on the right compare with those green spots on the left. Do we have a spot in the same place? And if we do, how does the fluorescence of that spot differ? And we've got 44,000 of them to check and test. Okay? So to do that, we use this analysis called gene spring, because of course we couldn't sit through 44,000 of these and test these by eye and do some sort of a measure. So we use this analysis called gene spring, and this actually compares very quickly the spot and how green it is, and compares it between the healthy and the control animal. Sorry, the healthy and the animal with pneumonia. And that allows us then to measure differential gene expression in pigs, in this case in pigs with pneumonia versus the healthy herd control. And what we do is we use this analysis called PCA, Principal Component Analysis. And this actually tells us, it's mathematically quite complex, but it tells us where covariance lies between the two groups. And it can measure masses of different variants, and then look at each one of those, where they compare and where they differ. And essentially, the closer these are together, so if we had healthy there in blue and pneumonia there in red, the closer they are together, the less differences there are between the genes in these two genomes. The further they are apart, obviously, the more difference there is between the two. So we can then start to say, well, they're far apart, so therefore there must be a lot of differences between the genes expressed in an animal with pneumonia versus a healthy control. In this case, what we found was that in the animals suspected with pneumonia, 1,837 genes were upregulated greater than twofold. Now, in these studies, a greater than twofold expression works out as 0.05 probability, 95% confidence limit. So anything which is greater than twofold, we would say is a significant difference. And so in this case, we had 1,837 genes which were significantly upregulated in the pigs with pneumonia. We also had 555 genes which were downregulated in pigs with pneumonia, significantly downregulated in pigs with pneumonia as well. So when we're looking at this, it's just as important to look at any genes which are downregulated as genes which are upregulated, because that can have as much of a bearing on what happens during production disease. So essentially we've got over 2,000 genes here where there's differential expression. Now, what we then did was to do a pathway analysis. Now what you can see from this slide is it's very blurred. And it's meant to be very blurred because what we've got is multiple pathways just piled on top of each other. So we can have, if you've got 2,000 genes and we look at the different pathways involved and connections between them, it's massive. But again, we've got software which can handle that as well. So using the Ingenuity pathway analysis, what we found was in these genes with pneumonia, 77 gene ontology pathways were affected in total. So out of all of those genes which were affected, these connected into 77 different pathways. And you can see what those different pathways are. I'm not going to talk too much about them, but we've got B-cell differentiation, adaptive immune response, and cellular immune response to external stimuli. And you can see all the genes there which are involved in this pathway, which were altered when the pig had pneumonia. So then what we need to do then is to narrow the gene profile if we want to determine potential biomarkers of production disease, and that's one of the things at Nottingham which we are actually interested in. What we really want to do is to take a massive gene set, narrow it down into smaller numbers of genes, and say when those genes change, that's when we see the production disease. So we get around the idea of having to culture microorganisms, which are difficult, the serological problems that I talked about, which are difficult, using cytokines as a biomarker, which hasn't really done much yet. It gets around all of those, and that's what we're really trying to do. So if we want to narrow the gene profile down to get to some sort of biomarker, what we then did was to compare the gene expression of pigs in that first study, the pigs with pneumonia, with pigs in another study. So we had pigs failed to thrive in housing with poor control of temperature and airflow, and pigs which were kept in unhygienic conditions. So if we look at upregulated genes, the first one is on the left, poor control temperature and airflow versus the pneumonia, the first one I talked about. Poor control of temperature and airflow, 317 genes were upregulated. Remember, there was 1,837 genes upregulated in pneumonia. But of these two data sets, 61 genes were upregulated in both. If we then look at genes which were downregulated, on the left there, poor air control, temperature, airflow, 592 genes were downregulated, 555 were downregulated in pneumonia, but only 14 genes were shared amongst these two data sets. So of all of these different genes in different variables, essentially, we've got a group that we know have pneumonia. We have a group of pigs which aren't thriving very well, and there's poor temperature control and airflow. We've narrowed this down now to say that there's 75 genes which are downregulated, but there's 75 genes in all of that data which appears to be the same. Shared. Upregulation, downregulation. So then what we did was we looked at pneumonia, upregulated pneumonia and temperature, and then we compared this again with poor hygiene. What we found with there's a slide missing here, but essentially what we found was that when we compared all of the 3 data sets, there was essentially 4 genes that we came to which were upregulated or downregulated, only 4 of them. In these massive 3 different groups, massive data sets, at the level of significance that we have set the bar at. What I should say is you can set it higher or lower and you will lose genes or obtain genes, but we set it at quite a strict level. We actually set it above 0.05, and when we did that, we had 4 genes. Okay, so we've now identified 4 genes, which we think then could be possible biomarkers. What we now need to do, and we're doing this as I speak, is we're going back to PCR. We take these 4 genes and then we use a PCR analysis of just of those 4 genes to, number 1, validate the microarray data that we've got, but number 2, take them and use them against tissues that we haven't actually microarrayed yet. So we have a lot of archived tissues, and we can then say, which we in some cases know the status of in terms of infection. We know that they have pneumonia or whatever. We can then PCR them and say, do these 4 genes still appear? And it's not important if they're upregulated or downregulated for a biomarker, it's just that they've changed. When we come to talk about, if we come to the right papers and we talk about the actual significance of these different genes, then it's important if they're upregulated or downregulated. But for a biomarker, we just need to know how they changed. And that's kind of where we're at the moment. Before I end, though, additional sort of results that we've had, which I think are quite interesting, is that when we've looked at intestinal tissue, so the animals had diarrhea and there's a production loss, and we looked obviously at the intestinal tissue, we see gene changes, but we then looked at the lung tissue as well, and we still see gene changes. Not the same, but we see gene changes there, and vice versa. So if the animal has had pneumonia and we look at the intestine, we see gene changes there as well, which I think is actually quite interesting, not so much probably from the point of view of a biomarker, but biologically, scientifically, I think that's quite interesting. Another thing that we found in many samples, in pigs and also chickens, is that many of these animals are immunosuppressed. So the immune profiles, the phenotypic profile of these animals is immunosuppression. And in some diseases, that's very, very significant. Now again, we haven't gone through all of this yet, but that could have real significance in that if we have animals that are kept in certain conditions and there's immune suppression, then obviously that would suggest that these animals then would be much more likely to come up to get a clinical infection with a pathogen. But it's possible that they're more likely to actually then get a poly-infection as well. So again, that could be important data that we sort of pick through in the future, which we're doing at the moment. So current and future work, so as I said, continue to analyse differential gene pathways recognised by microarray analysis, using construct PCR primers from common genes differentially expressed due to these conditions. We've already done that. And then take this down to a multiplex PCR analysis and go back to the archive samples. So essentially then what we've been trying to do is to look at production disease and say genomically we've got these massive number of genes and these different reasons why we've got production disease. Is there anything common about, any common characteristic there that we can actually really focus down, and instead of having to look at massive genes, we can just look at a handful of them and say that is then that's what a production disease looks like. Other things that we want to do though is that if we then say this is what pneumonia looks like, well, what is pneumonia? What sort of levels of pathogen do we need? What pathogen do we need? Is that shared? When we diagnose this, can we diagnose it immediately, or does it have to be progressed to a certain level before we can do that? So there's a lot of work still to be done with this, but what we've done here is we've took the big data set and kind of got it to a level where at least we can now ask those questions, hopefully. Okay, so this is most of us, this is the main group who's worked on this. We have Tim Giles on the left, Scott Hume, Paul Barrow, and me. And thanks for listening. Thank you.